Hello students, I am Shahin Mahmood studying MBBS from College of Medicine and JNM Hospital Kalyani, and I will explain the botany section of FL5 starting with the first question. Which of the following statement about five kingdom classification is incorrect? So we need to find the incorrect statement about five kingdom classification. First statement is given, R.H. Whitaker proposed a five kingdom classification. We know that there were several kingdom classification like two kingdom classification, five kingdom classification, as well as there were six kingdom classification, out of which five kingdom classification was proposed by R.H. Whitaker. So this is the correct statement. Next statement is given, all the unicellular eukaryotic organisms are grouped together under kingdom Monera. So we know that Monera is itself a prokaryotic group. So unicellular eukaryotic organisms, this statement is itself incorrect. So unicellular eukaryotic organisms are grouped under kingdom Protista. Next, the five kingdom classification was proposed in 1969. The five kingdom classification which was proposed by R.H. Whitaker, it was given in 1969. This is also a correct statement. The next statement is saying the main criteria for classification of the kingdom include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Let's check if all are correct. So the main criteria for classification of the kingdom, cell structure is right, body organization is also right, mode of nutrition is also right, reproduction and phylogenetic relationship. So all the things can be considered criteria for classification of this kingdom. So this is also a correct statement. So we need to find the incorrect statement, and we can see that second statement is incorrect. So second will be our correct answer for this question. Second option. Now moving to the next question. So it is saying, which of the following are found in extreme saline condition? Eubacteria, cyanobacteria, mycobacteria, archaebacteria. So we know that archaebacteria have a type that is called halophiles. Halophiles. So halophiles are the bacteria, are a kind of archaebacteria which can withstand high temperature and that can survive in salt pans, marshy areas, salty areas, salt beds, etc. So archaebacteria is the correct answer. And that why they can withstand high temperature or extreme saline condition, because they have a different cell wall structure. Rest other options are given, eubacteria, cyanobacteria, mycobacteria have not a special cell wall structure. So fourth option will be our correct answer for this question. Now moving to the next question. 103. Which of the following statement about mycoplasma is wrong? They are pleomorphic. We know that pleomorphism means they have no definite shape or size or structure. So yes, mycoplasma, we need to understand the features of mycoplasma. So mycoplasma is small, unicellular, non-motile, prokaryotic organism. So they are pleomorphic, and because of this pleomorphism, they are also known as pleuropneumonia-like organisms. So let's start that. Let's read the statements. They are pleomorphic. This is a correct statement. Next statement is saying they are sensitive to penicillin. So we know that penicillin acts on cell wall, and this mycoplasma lacks cell wall. That's why penicillin can't act on them. So penicillin, they are not sensitive to penicillin. Though this is a wrong statement. Next statement, they cause diseases in plants. So obviously mycoplasma can cause diseases in plants. So this is a correct statement. Next, they are also called PPLO. So PPLO, the full form of PPLO is pleuropneumonia-like organism. So this is also a correct statement. So we need to find the wrong statement. Second option is wrong. So second option will be our correct answer for this question. Now moving to the next question. 104. So it is saying, which of the following accurately describes the pigments of the members of Chlorophyceae? First of all, we need to find what are the pigments present in Chlorophyceae. So we know that the pigments present in Chlorophyceae are chlorophyll A and chlorophyll B. And because of this pigment, they are greenish in color. So they are green in color. So now read the options. They are primarily red pigment, which is incorrect. They have predominantly blue pigment, which is also incorrect. They have mainly green pigment. This is a correct statement. They have primarily yellow pigment, which is also not correct. So third option will be our correct answer for this question, because of the dominance of the pigment chlorophyll A and B. Members of Chlorophyceae have green pigments. Now moving to the next question. What is the eye of potato? What is the eye of potato? Suppose this is the potato. We know that potato is the underground vegetable. You can see there are several kinds of this thing on the surface of potato. So what are these? So basically, these are called in potato. These are called axillary bud. So these are called axillary bud. Axillary bud is called tuber. So what is a tuber? A tuber is basically a solid tip of underground branch, and tubers could be round or oval in shape, and each tuber have many notches on the surfaces as we can see in this picture. These are called eyes. So basically, first option will be our correct answer for this question. Now moving to the next question. Axial presentation is presenting. So first of all, we need to understand what our axial presentation can be, how axial presentation can be described. So axial presentation can be described when the placenta is axial and the ovules are attached to it in a multilocular ovary. So the presentation is said to be axial. Suppose this is, you can see these are the presentation. Suppose presentation multilocular, we have to find out. This is a kind of axial presentation. You can see multilocular ovary. So these are found in China rose, tomato and lemon. So P is not a correct answer. Argemone is not a correct answer. Dianthus is not a correct answer. Lemon is the correct answer. So fourth option will be our correct answer for this question. So now moving to the next question. Potato and sweet potato. So we need to read the statements and tell about which is the correct about this potato and sweet potato. Have edible parts which are homologous organs. So basically, what does homologous organ means? So homologous organ means they have the same origin but different functions. So it is not correct. Now they have edible parts which are analogous organs. Analogous organ means they have different origin but serve the same function. So it is absolutely correct. Now the next statement is saying have been introduced in India from the same place. It is also not correct. Are two species of the same genus, which is also not correct. So second option will be our correct answer for this question. That potato or sweet potato are analogous organs. So moving to the next question. 108. Yeah. So the aleurone layer in maize grain is especially rich in proteins, starch, lipids, or oxygen. So we need to find the aleurone layer. So first of all, maize grain is a monocot. Maize is a monocot. Monocotyledonous plant. Monocot. So we know that monocot, if you see the of monocot plant, the somewhat look like if you try to make a diagram. So we'll see that there is a certain kind of layer will be there. So this is called aleurone layer. Aleurone layer. So the aleurone layer. So this basically aleurone layer is basically made up of proteins. So first option will be our correct answer for this question. It is made up of proteins, not starch, not lipid, or not oxygen. Oxygen is basically a plant hormone. Now moving to the next question. That stomata are the structure present in the epidermis of leaves, and the reason is given. Stomata regulate the process of transpiration and gaseous exchange. We know that stomata in leaves, suppose this is a leaf. So in leaves, this upper part and lower part. We know that stomata is present uniformly, like not uniformly, but all parts of these leaves, but more in the lower part of this leaves, lower epidermis, more than upper epidermis. Lower epidermis, more stomatas are present. The statement is here, stomata, the structure present in the epidermis of leaves, is definitely correct. And the second statement is stomata regulate the process of transpiration and gaseous exchange, which is also correct. Now we read the statement. Assertion is correct, but the reason is incorrect. It is not correct. Assertion and reason are correct, but reason is not the correct explanation of the assertion. This will be the answer, because we know that stomata are the structure present in the epidermis of leaves, and these are not the solely purpose of the stomata. Stomata can act like stomata have various functions beyond transpiration and gas exchange, including the regulation of water loss and the exchange of gases necessary for photosynthesis. So basically this will be our correct answer for this question. Second option. Assertion and reason are incorrect. This is also not correct. Assertion and reason are correct, and reason is the correct explanation of the assertion. It is also not correct. Now moving to the next question. Below are the two given statements. Statement 1. In dicotyledonous stems, cambium is present between phloem and xylem. This is absolutely correct statement. And the next statement. In the monocotyledonous stem, cambium is present between phloem and xylem. We know that in monocotyledonous plants, cambium is not present. So basically, this is an incorrect statement. So the first statement is true, but second statement is false. This is the correct answer itself. First option is the correct answer for this question. Moving to the next question, 111. It is saying a special membranous structure is the mesosomes, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of. So basically, we will draw a structure. So basically, we will understand what mesosome is. Basically, this is a bacterial cell. Trying a mesosome. Suppose this is a cell wall. This is a cell membrane. And this is the extension of cell membrane. We can say this is mesosome. So basically, these mesosomes are in the form of vesicle, tubules, and lamellae. So we need to find these options in this, and we can see that in fourth option it is given that the extensions are in the form of vesicle, tubules, and lamellae. So fourth option will be our correct answer for this question. Now moving to the next question, 112. Which of the following statement about the ribosomes and inclusion bodies is incorrect? You need to find the incorrect statement. So the first statement is given. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. This is the correct statement. Next statement is given. Ribosomes are the site of protein synthesis. Ribosomes are the site of protein synthesis. This is also a correct statement. Next statement is given. Inclusions bodies are not bound by any membranous system and lie free in cytoplasm. This is also right, because if this is a cell, a bacterial cell, then we can see the inclusion bodies are not membrane bound and this is lie free in cytoplasm. Now the next is given that in prokaryotes, ribosomes are of two subunits, namely 30 and 40S. We know that in prokaryotes, ribosomes are 50S and 30S, and they both together comprises 70S subunits. This is the incorrect statement, and we need to find the incorrect statement in this question. So fourth option will be the correct answer for this question. Fourth option. Now moving to the next question. Rubisco has much greater affinity for O2 than CO2. The next statement, RUBP binds with O2 to form two molecular phosphoglyceride. So first verify the first statement. Rubisco has a much greater affinity for O2 than CO2. So Rubisco basically has a much greater affinity for CO2 when the CO2 and O2 concentration or this is equal. CO2 and O2 concentration basically equal, then they have much more affinity towards CO2, not O2. So this is the incorrect statement. Next statement, RUBP binds with O2 to form two molecules of 3-phosphoglyceride. We know that this happens in C3 cycle. But RUBP doesn't bind with O2, it binds with CO2. So RUBP binds with CO2, then it forms 3-phosphoglyceride as the first stable product of C3 cycle. So basically this is an incorrect statement. So both the statements are incorrect. So second option will be our correct answer for this question. Moving towards the next question. At oxaloacetic acid is the first stable product of C2 plants. No, C2 plants no. C4 plants is the correct answer, because we know that C3 plants have the PGA, phosphoglyceric acid, as the first stable product. So third option will be our correct answer for this question. Now moving to the next question. The most striking example of point mutation is found in a disease called. So we need to find among this all options. Down syndrome, we know that Down syndrome basically trisomy of chromosome. Trisomy of chromosome 21. Sickle cell anemia is the example of point mutation that involves only the replacement of one nucleotide with another. So this is an example of point mutation. However, what happens that GAG is replaced by GTG. So GAG is coded for glutamic acid. Now GTG is coding for valine. So this is a point mutation. So this is the most striking example of point mutation. Second option will be your correct answer. Klinefelter syndrome is also a chromosomal disorder, and night blindness is a vitamin deficiency. Vitamin A deficiency. Now moving to the next question. Who argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried? So basically, this question saying chromosomal segregation, and the chromosomal theory of inheritance was proved by Sutton and Boveri. So Sutton and Boveri will be the correct answer for this question. T.H. Morgan is linked with linkage, and Sladen and Schwann is linked with cell theory. Corona and Schwann is not relevant in this question. So moving to the next question. Statement 1. DNA being double-stranded resists the changes by evolving the process of repair. Statement 2. A segment of DNA that codes for RNA can be referred as gene. So you know that DNA are double-stranded. Somehow we made it. This is not actual representation. So DNA somehow look like this. So first statement we will try to verify. And DNA being double-stranded, we can see two strands of DNA. So because of this double-stranded, they are having a complementary strand. So that's why they can resist the changes by evolving the process of repair. So this is absolutely correct statement. The next statement is saying a segment of DNA. Suppose this is a segment of DNA. This is a segment. This segment of DNA that codes for RNA. We know that DNA, central dogma, DNA to RNA, and then RNA to proteins. So that is the central dogma. So this segment of DNA that code for RNA is referred as gene. So this is absolutely correct statement. So both the statements are correct. So first option will be our correct answer for this question. You can see that somehow that so many statement questions are coming. So you need to read the NCERT more and more. You need to revise more and more so that you will not miss this kind of questions. Now moving to the next question. Which of the following codon has dual function? So basically these are the codons given: CGG, AUG, UGA, UC. We know that AUG serves as initiator codon. Initiator codon. It also codes for methionine. So it acts, or it is having dual nature, dual function. So this will be our correct answer for this question. UGA is a stop codon. The second option will be our correct answer for this question. Now, next question. For the next question, 190. Which of the last human chromosome that completely sequenced? Chromosome 1, chromosome 11, chromosome 21, chromosome X. You know that chromosome first is the last human chromosome to be completely sequenced. So this was completed only in May 2006. And this is the last chromosome to be sequenced. So first option will be our correct. So this is basically a direct question from NCERT Human Genome Project. So you need to read NCERT. So 120. Next question. Saccharide groups are especially useful in generating environmental awareness, preventing soil erosion, year-round flow of water in rivers, conserving rare and threatened species. So So we need to understand what sacred groups are. So basically sacred groups is an example of in situ conservation. Basically in situ conservation. Right? Something is conserved at that place itself, not taking in the outside environment. So conservation of forest and wildlife, especially rare and threatened species. So this the fourth option will be our correct answer for this question. So these are found in places of worship which are held in high esteem by tribal communities. They are most undisturbed forest patches which are often surrounded by highly degraded landscapes. Such sacred groups are found in Kashi and Jaintia hills of Meghalaya, Aravalli hills in Rajasthan, Western Ghat regions of Karnataka. So basically these are the examples. Moving to the next question, 121. So as we go from species to the kingdom, the taxonomic hierarchy, the number of common characteristics will basically decrease. First question will be your correct answer. Now we will understand what will happen. So during my time, I used to remember a trick that tracking while I was attempting for NEET, KPC of GS. So if you remember, K stands for kingdom, P stands for phylum, E stands for class, O stands for order, F stands for family, G stands for genus, and S stands for species. So basically when you go from up to down, you will see the characteristics among the organism increases, and when you will go from like hierarchy, you will go from down to up, you will see that the more number of characteristics decreases. So in this question saying, as we go from species to kingdoms, we are going down to up. So basically in taxonomic hierarchy, the number of common characteristics will definitely decrease. The first option will be our correct answer for this question. First option. Not increase, not remains, may increase or decrease, not a correct answer. So the next question we will move. 120. Match the following. So we just need to match the following. This is the complete questions, like a table question from biomolecules. So we need to remember each and every table that NCERT provided to remember. So the first, vinblastine. So vinblastine is basically a drug. Vinblastine is basically a drug. Now the second is reaching. So reaching is basically a kind of toxin. Now codeine. So codeine is basically an alkaloid, and gum is here basically a polymer. So you can see that our matching is completely matched with option number third. The third option will be your correct answer for this question. Going to the next question. So almost all enzymes are proteins and are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituent called as apoenzyme, cofactor, allosteric coenzyme. So you have to understand this. So suppose this is the structure of enzyme, and these are enzymes. These have made up of polypeptide chains. So these are polypeptide chains. As well. Now, if we add these two things, these are called cofactors. So they basically act as catalyst. They will enhance the activity of enzyme, and they are non-proteinaceous. They are non-protein structure. So basically these are called cofactors. This will be our correct answer for this question. Second option will be our correct answer. Now moving to the next question, 134. So the characteristic feature of diplotin stage of meiosis is recognized by dissolution of synaptonymal complex, homologous chromosomes of bivalent start separate from each other, and then X-shaped structure chiasma are formed. This event described here in the diplotin stage of meiosis for prophase 1. So these are basically all are correct. So fourth option will be our correct answer for this question. Now moving to the next question. Terminalization, synaptonymal complex, chiasma are the terms basically related to. So these are basically related to the meiosis first. We know these are seen in prophase 1. So meiosis 1 will be our correct answer for this question. More specifically, in diplotin stage of prophase 1. So now we'll move into the next question. 126. Given below are two statements. Statement 1. During oxidative decarboxylation of one molecule of pyruvic acid in mitochondrial matrix, one molecule of NADPH plus H plus is produced. And the next statement is saying, in glycolysis, glucose undergoes partial oxidation to form two molecules of pyruvic acid. So now we will verify the first statement. During oxidative decarboxylation of one molecule of pyruvic acid in mitochondrial matrix, one molecule of NADH is produced, not NADPH. This is basically a wrong statement. Next statement is saying, in glycolysis, glucose undergoes partial oxidation to form two molecules of pyruvic acid. This is the correct statement. The first statement is correct and second statement is incorrect. So that's why second option will be our correct answer for this question. First option will be, second option is not correct because the first statement is wrong. Third option will be our correct because the first statement is incorrect, but the statement second is correct. Now moving to the next question. 127. Which of the following molecules are utilized as well as released during glycolysis process? So if we see glycolysis, we will see that ATP is being used as well as released in the whole process of glycolysis. H2O is being released, or H2O are released during the conversion of 2-phosphoglycerate to 2-phosphenol pyruvate, but nowhere it is utilized. So basically second option will be our correct answer for this question. And H2O, CO2 basically these are irrelevant because nowhere these are getting, sorry, CO2 and O2. CO2 and O2 are basically irrelevant because nowhere in glycolysis they are being utilized or released. Now moving to the next question. 128. The simplest expression of arithmetic growth is exemplified by a root elongation at a constant rate. This curve from this kind of growth is basically a linear curve. The second option will be our correct answer because the growth of root according to the growth. The growth of root is basically a constant growth. Sigmoid curve we know basically this kind of curve, and the parabola is basically this kind of curve, and parallel curve is not relevant here. Going to the next question. It takes very long time for pineapple plants to produce flower. Which combination of hormone can be applied to artificially induce flowering in pineapple plants throughout the year to increase the productivity? So basically we need to apply what are the combinations of hormone to induce flowering in pineapple plants. So that is basically auxin and ethylene. So first option will be our correct answer. If we talk about other options, gibberellin and cytokinin is not a combination. Gibberellin and abscisic acid is basically a senescence hormone. This is also not a correct option. So abscisic acid where placed in the option, that would not be definitely the answer. Now moving to the next question. A patient brought to hospital with myocardial infarction in January. He is treated with cyclosporin A, statin, penicillin, or streptokinase. We know that cyclosporin A is basically an immunosuppressant. Now, statins are basically like hypocholesterol agent, cholesterol lowering agent. Penicillin basically antibiotic. Streptokinase is a clot buster. So it will remove clots. It helps to remove clots from the blood vessels of the patients who have undergone myocardial infarction leading to heart attack. So fourth option, streptokinase, will be a correct answer for this question. Now moving to the next question 131. Given below are two statements. Ladybird and dragonflies are useful to get rid of mosquito and aphids respectively. Statement second, Trichoderma species are effective biocontrol agent for several plant pathogens. Now, ladybird and dragonflies are useful to get rid of mosquito and aphids. It's not a correct statement. This will look similar because this exactly one thing is twisted here. So, ladybird is basically to get rid of aphids, and dragonfly is basically to get rid of mosquitoes. So, it is just twisted here. So, you need to take care of that. That's why the statement is wrong. Now the second statement, Trichoderma species are effective biocontrol agent of several plant pathogens is absolutely correct statement. So the third option, statement 1 is incorrect and statement second is correct. Will be our correct answer for this question. Now moving to the next question, 132. There is a special phenomena found in Astraceae family, which is a type of asexual reproduction that mimics sexual reproduction. Which among the following is that? Syngamy, apomixis, polyembryony, or parthenogenesis? We know that all those seeds in general are the products of fertilization. The seeds basically are the products of fertilization. A few flowering plants, such as the family of Astraceae, what they have, and this specifically, if you wanna mention in Astraceae family, it's grasses. So grasses have a certain kind of thing that have evolved a special mechanism to produce seeds without fertilization. Seeds are producing without fertilization. So that mimicking the sexual reproduction. This is basically asexual reproduction, but that mimicking sexual reproduction because they are producing seeds. So this process is known as apomixis. The second option will be your correct answer for this question. Now moving to the next question. The gynoecium consists of many free pistils in a flower of apophis, michelia, aloe or tomato. If you see the structure or the diagram given in michelia, for michelia in NCERT, some sort of this kind of diagram is given here. So this is basically a multicarpellary apocarpous gynoecium. Second option will be our correct answer for this question. This is multicarpellary. Moving to the next question. 134. Which of the following population interaction is widely used in medical science for the production of antibiotics? So basically, for production of antibiotic, what happens? That one is getting harmed. One is getting harmed, that is bacteria. And one is getting no benefit. This is also a bacteria. But the thing is, this bacteria is disease causing, this is harmful, and this bacteria is not harmful. So this is the case of amencellism. Fourth option will be our correct answer for this question. Now moving to the next question. 135. If we completely remove 135, let's read out now. If we completely remove the decomposers from an ecosystem, its functioning will be adversely affected because mineral movement will be blocked. We know that decomposers help in the movement of minerals, basically. And we'll just read out other options, then we'll decide if any better options is given. So don't just blindly tick out that first option you read that this is matching and you just tick that. No, don't do that. Read all the options. The rate of decomposition will be very high. So if my decomposers are removed from ecosystem, the rate of decomposition will be very low, not high. Energy flow will be blocked. Energy flow will be not blocked because of the decomposers, because decomposers not coming to display. Herbivores will not receive solar energy. So herbivore will definitely receive solar energy. That doesn't depend on decomposers. So the first option, mineral movement will be blocked, is the correct answer for this question. Now moving to the next section, section B. So first question of section D is a statement based question. So it is saying below are the two given statements. Statement 1, chemosynthetic autotrophic bacteria oxidizes various inorganic substances such as nitrates, nitrites and ammonia. Second statement is saying eubacteria are characterized by the presence of a cell membrane and if motile a flagella. So we need to verify. Chemosynthetic autotrophic bacteria oxidize various inorganic substances, nitrate, nitrites, ammonia. This is absolutely correct statement. They oxidize their substances and use their released energy for their ATP production. Now the next statement, eubacteria, also known as true bacteria, characterized by a presence of cell membrane. Basically, eubacteria doesn't have a cell membrane, it is characterized by a cell wall, and by a presence of a rigid cell wall, and if motile, a flagella. So this is a wrong statement. So we can say that the first statement is true, but second statement is false. First option will be our correct answer. Going to the next statement. Next question. Match the column. Column 1 with column 2. Chlorophyce, pheophyce, and then rhodophyce. So to solve this question, I will use a simple trick here. So the trick is simple. CPR. We need to chlorophyce, pheophyce, rhodophyce. And we know that chlorophyll A in all this family have chlorophyll A. So we'll write chlorophyll A in all these. And then B, C. Now match. Chlorophyce have chlorophyll A and B. So chlorophyce have chlorophyll A and B. Pheophyce have chlorophyll A and C. So B will be matched with. So this will be matched with A and C. And now rhodophyce is matched with chlorophyll A and B. So here we can see 137. So chlorophyce have chlorophyll A and B, pheophyce have chlorophyll A and C, and rhodophyce have chlorophyll A and D. By using the simple trick, you can just mark this answer. Second option will be our correct answer for this question. Now moving to the next question. Consider the following statements regarding the life cycle of bryophytes. The anthrazoids are released into water while they come in contact with archegonia. This is a correct statement. Why it's a correct statement? Just think that water helps air for the contact. Now the second statement is given. The anthrazoid fuses with egg to produce zygote. This is also a correct statement that the anthrazoid of bryophytes fuses with the egg to produce the zygote. Now zygote undergo reduction division immediately. No, this is a wrong statement. A zygote doesn't undergo reduction division immediately. They produce a multicellular body known as a sporophyte. This is a wrong statement. How many of the above statements are false? So one statement is false. So the first option will be our correct answer. Now moving to the next question. 139. So when a shoot tip transforms into a dash, it is always dash. The arrangement of flowers on the floral axis is termed as. So here comes the reading of NCERT. Immense reading of NCERT. If you revise multiple times the line, this is basically a question from lines of the NCERT. If you read the NCERT multiple times, then you can solve this question. Otherwise, you can't. So basically, I will advise to read NCERT multiple times as much you can. So now we solve the question. When a shoot tip transforms into a flower, shoot tips transforming into a flower so that it is always solitary. So if shoot tip transform into a flower, it gets solitary. And the arrangement of flowers, just try to understand, arrangement of flowers on the floral axis is termed as inflorescence. The third option will be our correct answer for this question. Now moving to the next question. Precentral placentation is found. First of all, we will try to understand what does precentral placentation means. Basically, free central concentration means that ovules are free and trays are not bound by any wall. So we'll try to make some sort of structure. So trays are basically ovules, and this is placenta. This is free central concentration. You can see that ovules are not bound by any kind of ovary walls. So this free central concentration is found in dianthus. First option will be your correct answer. Moving to the next question. 141. Below are the two given statements. The outermost layer is epiblema in a dicot root, and the cortex consists of several layers of the thin walled parenchyma cells with intercellular spaces in dicot root. So we will try to visualize the structure of dicot root here. So this is how we can make. Just make some. Suppose this is the structure of dicot root. We just made the initial parts. So this outermost layer will be called as epiblema, and this is, suppose this part is the cortex. So this will consist of several layers, and this would have thin walled parenchymal cell with intercellular spaces. This is the structure of dicot root. So we can see that our statement, both statements are true. So third option will be our correct answer for this question. But this is applicable for dicot root. Now moving to the next question, 142. It is saying the cell membrane is mainly composed of lipids and proteins, and the reason is saying the major lipids in the cell membrane are phospholipids arranged in a bilayer with a polar heads facing outward and hydrophobic tails facing inward. So when you are getting confused this kind of question, just put like that the cell membrane is mainly composed of lipids and protein because. Just put here, now read. Because the major lipids in the cell membrane are phospholipids arranged in a bilayer with polar heads facing outwards and hydrophobic tail facing inwards. This statement is also correct. This is also correct. But the thing is that when you are putting because here, you can see that this here described that cell membrane is mainly composed of lipids and protein, but nowhere in this reason there is a mention of protein. So this statement is also correct, but it doesn't describe the whole composition of the cell, which include both lipid and protein. It is discussing only the lipid part only. So no protein, the structure of protein is not described here. So the reason is not satisfied the assertion. So what will mark? Second option. The assertion and reason both are correct, but reason is not the correct explanation of the assertion. Second option will be our correct answer for this question. Now moving to the next question. 140. That which of the following organelle is enclosed by a single membrane: lysosome, nuclei, mitochondria, chloroplast. Nuclei, mitochondria, and chloroplast all have double membrane. There is a membrane bound organelles, double membrane bound organelles. And lysosome, so basically lysosomes are a small vesicle bounded by a single membrane, and this contain hydrolytic enzymes. So first option will be our correct answer for this question. Moving to the next question, 144. That a graph plotting light absorption by pigments in a single wavelength is called absorption spectrum. This is a correct statement. The graph showing the rate of photosynthesis at different wavelengths of light is called action spectrum. So it is saying that the curve showing the rate of photosynthesis at different wavelengths of light is called action spectrum. This is also a correct statement. It resembles the absorption spectrum of chlorophyll A and B. Both statements are correct. The first option will be your correct answer for this question. Now moving to the next question, 145. In crunch anatomy, the bundle sheath cells have. Which plants have crunch anatomy? C4 plants have crunch anatomy, and the bundle sheath cell of C4 plants What are the features of the bundle-shaped cells of C4 plants which have branch anatomy? So these are basically thick cells, thick kind of cells, and it also that it forms several layers around vascular bundles. So they have large number of chloroplasts, thick walls. Why they are thick walls? Because it is impervious to gaseous exchange and they have no intercellular spaces. So the features are thick walls, no intercellular spaces and large number of chloroplasts. So second option will be our correct answer for this question. You can see the next, moving to the next question. 146 statement based question. Which of the following statement is incorrect? Mendelian disorders are mainly determined by alteration of mutation in a single gene. Second statement, sickle cell anemia. Let's prove one by one. Mendelian disorders are mainly determined by the alteration of mutation in the single gene. This is absolutely correct statement. This is how Mendelian disorders are described. Sickle cell anemia is an autosomal linked recessive trait. This is also a correct statement, sickle cell anemia. And now that failure of segregation of chromatid during cell division cycle results in the gain or loss of a chromosome is called aneuploidy. So if you want to understand this statement, this is the chromatid structure. So if this failed to segregate while cell division, then this got maybe gain or maybe lose. So this phenomena is called aneuploidy. This is also a correct statement. Now it is saying, fourth statement, Turner syndrome, Klinefelter syndrome are common examples of Mendelian disorder. This is not a correct statement because Turner syndrome and Klinefelter syndrome are basically chromosomal disorder. Chromosomal disorders are not included in Mendelian disorders. So third option will be your correct answer. This is saying the incorrect option we need to find out. So third option will be your correct answer in this question. Now moving to the next question. 147. That is basically you can see which of the following, which one from those given below is the period for Mendel's hybridization experiment. So you need to remember this kind of question. You need to remember the year which you worked on. That was basically 7 years. So you can see that in this option, if you are not remembering that 7 years, you don't know which specifically what year it was. You can see this was 10 years. This could not be the answer. You can see also this exceeds. This basically exceeds more than 7 years. This also could not be the answer. 70 to 77. So the confusion lies between these two. This is also 7 years. This is also 7 years. So 1856 to 1863 is the correct answer for this question. So you need to remember the year for Mendel hybridization experiments. Now going to the next question. 148. Match the list 1 with list. We have to basically the features of DNA are given. So we will basically make DNA here, and now we will try to compare. This is a rough structure. Now read out the statements. List 1 first. Diameter of DNA helix. This is basically the diameter of the DNA helix. This is the diameter of DNA helix. So this is the measurement is 20 angstrom. 20 angstrom here. The diameter of DNA helix is 20 angstrom. Now the distance between two adjacent base pair. So this is the distance between two adjacent base pair. So this is basically 3.4 angstrom. 0.4 distance between two base pair. This is one base pair, second base pair. Distance between two base pair. Now the pitch of DNA helix. So this is the pitch of DNA helix. You can see this is the pitch. Sorry. This is the pitch. So this comprises 3.4 nanometer, and base pair in nucleosome is 200. So to match this, base pair in nucleosome is 200. If we match this, you can see second option matching. all our list 1 and list 2. Second option will be your correct answer for this question. Now moving to the next question, 149. DNA fingerprinting involves identifying differences in some specific regions in DNA sequences called as repetitive DNA. In repetitive DNA sequences, a small stretch of DNA is repeated many times. So we know that in human DNA, 99.99% specifically 99.9% of human genome or DNA are similar. So what happens that this sequence normally do not code for proteins, but they form a large portion of human genome. This sequence have show high degree of polymorphism. High degree of polymorphism. High degree of polymorphism and form the basis of DNA fingerprinting. As these polymorphisms are inheritable from parents to children, so DNA fingerprinting can be used for many purposes like basis of paternity, testing, in case of discourse. But in this question, it is saying that if we want to verify assertion with reason, in repetitive DNA sequences, a small stretch of DNA is repeated many times. DNA fingerprinting involves identifying differences. Why this is getting differences? Because these are getting repeated. A small stretch is getting repeated many times. This is absolutely correct. And both assertion and reason are true. So first option will be our correct answer for this question. Now moving to the next question. 150, which organization publishes the Red Data Book? Red Data Book published by IUCN. What is the full form of IUCN? International Union of Conservation of Nature and Natural Resources. So second option will be our correct answer for this question. This concludes the explanation of FL5 of Botany.